Good morning, welcome Sham Stars. As in previous video, we have already discussed about mega project, that is Human Genome Project. So today we are going in the continuation of the previous video, that is methodologies, salient features of Human Genome Project and applications. Let's talk about the methodology. The methods involve two major approaches. One approach focused on identifying all the genes that are expressed as RNA. In our DNA, there are two parts, coding parts and non-coding parts. So coding parts, it translates or codes for protein synthesis. So the first approach was to code or to sequence only coding part of the DNA. This methodology is termed as express sequence tags or ESTs. They plan to sequence only the DNA part which codes for protein. Other approach was to sequence entire DNA. It means coding and non-coding part of the DNA, and it was followed, and it is called sequence annotation. Let's talk about the ESTs, expressed sequence tag. It means the coded genes which code for the RNA, and their use was promoted by Venter in 1991, and it was a revolutionary way for gene identification. They are unedited, randomly selected single-pass sequence reads derived from the DNA libraries. So the second approach, which was the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence, and later assigning different regions in the sequence with functions, a term referred to as sequence annotation. Annotation means process of identifying the location of genes and all the coding regions in a genome and determining what are the functions of those genes. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively small sizes with the help of endonucleases and cloned in suitable host using the specialized vectors. The cloning results into the amplification of each piece of the DNA fragment so that it subsequently could be sequenced with ease. The commonly used host were bacteria and yeast, and the vector used, we call it BAC, bacterial artificial chromosomes, and YAC, yeast artificial chromosomes. Now let's talk about salient features of human genome. The human genome contains 3164.7 million nucleotide bases. Human genome contains the information of 24 chromosomes, 22 autosomes and X chromosome and 1 Y chromosome. The human genome contains over 3 billion nucleotide pairs. Human genome is estimated to have about 30,000 genes. Less than 2% of the genome codes for protein. The average gene consists of 3000 bases, but sizes vary greatly. Chromosome 1 has most genes, that is 2968, and the Y has the fewest, that is 231 genes. Scientists have identified about 1.4 million locations where single base DNA differences occur in human beings. This information promises to revolutionize the process of finding chromosomal location for disease-associated sequences and tracing human history. Now let's talk about applications of Human Genome Project. Identification of human genes and their functions, understanding of polygenic disorders, for example cancer, hypertension and diabetes, improvements in the gene therapy, improved diagnosis of diseases and development of pharmacogenesis. So children, this is all about Human Genome Project. I hope you must have understood. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.